The development of specific genes, by definition, is the regulated growth resulting from interaction of genomes in the environment, which results in a sequence of events that give a certain structure or show a specific phenotype for an organism. To explain how this works, we will look at an example of the development sequence of spikes of certain cells on a spiny beetle. Here the arrows represent activation of the gene, while the T-bar represents the inhibition of the gene. In order for these cells to show spikes, the S activates transcription of P. The product of P activates the transcription of I. The I product then inhibits the transcription of K, whose purpose is to repress the function of E. Since K can no longer perform this function due to I, then E is activated, and therefore we show the phenotype of spikes. Now, if we wanted to see how mutations affect this cascade, we can see how important these specific genes are to produce the desired phenotype. For the first mutation, if we were to lack the P gene, the I gene would not be activated, and thus the K gene would be activated and inhibit E, which inhibits the phenotype of spikes. If there was an I mutation and it was not present, a similar result would happen, that the K gene would activate and repress the E gene, and therefore no E gene would result in no spikes. If we were to have a W mutation where both I and E are removed, E is needed in order to produce spikes, so if we were to not have an E gene, there is no way we could produce spikes on a beetle. If we were to look at one last mutation, one where S and K are missing, S would not be able to activate P, and therefore P cannot activate I, which would inhibit K normally and prevent the phenotype of spikes. However, the K gene is also not present, which would prevent it from inactivating E, and therefore E is activated and we have the spiky phenotype. These mutations cause some cells to not have spikes in the ectoderm of beetles, and that is due to improper signaling and can have various effects like express variability or even no expression. There is also a lack of need for all cells in the ectoderm to be spiky. For example, we do not need neurons within our brain and other parts of the nervous system to be pointy, and also the outer layer of internal organs to be pointy and thus cause serious damage to other internal structures within our body.